In the second part, we'll talk some single cellular morphology and behaviors from both the intercellular structures and some extracellular structures. So the learning objectives of this part is we expect you could describe the common bacterial cell morphologies and understand how cells maintain their morphology. And you could be able to list at least 2 bacterial cytoskeleton proteins and describe their biological functions in general. Understand how the cytoskeleton and peptidoglycan control the cell shapes and the cell division. Be able to distinguish different arrangements of bacterial flagella, knowing the basic parts of bacterial flagellum and the flagellum assembling process. And you also expect to know the rotation of bacteria and the 10 tumble motilities. So just before we get into the details of intercellular structures of bacteria, I would like to spend several slides to quickly recall some knowledge from the previous lectures. So what we learned in the previous lecture is bacteria are not just little unorganized bags of proteins. Instead, they are small but sophisticated. As you can see from these slides, they have well-designed structures as shown. Well, also we talked a little bit about the 2 types of bacteria, which is gram-positive bacteria and gram-negative bacteria. So the main differences are their cell envelope structures. As you can see in these slides, the gram-positive bacteria, such as Bacillus, they have only 1 layer of cell membranes, while in the gram-negative bacteria E. coli, you can see that they have both inner membrane and outer membrane, 2 layers of cell membranes. Well, also Yan introduced you a little bit about the cell have evolved a wide variety of shapes from, you see, from the rod shapes of bacilli and also the round shape of Staph aureus. However, the evolution of complex shapes must be accompanied by the evolution of mechanisms to control the cell shapes. So the cell wall, especially the peptidoglycan layer, plays an important role in defining the cell shapes. Peptidoglycan consists of 1 single micromolecules formed glycan strands that are cross-linked by short peptides, as shown in the next slides. So once E. coli cells has 500 to 1,000 glycan strands on its long axis, while 70 to 120 glycan strands in their round surrounding cross sections. Of course, when bacterial cell grows, the peptidoglycan also grows. So the molecular mechanisms of peptidoglycan growth is a little bit complicated, but biological controlled enzymatic processes. So it requires both synthetic and hydrolytic reactions. So this table also shows you lists that more than 20 proteins that are known to involve in this process in E. coli. Well, this number can be increased in other bacteria with more complicated shapes. So what happens if there's a mutant in this gene? Well, as I introduced you a little bit in the previous slides, there's Campylobacter jejuni cells that has 2 distinguished colony morphologies. So when the researchers try to zoom in the bacteria from these colonies, they found actually they consist of two different shapes of bacteria. So the difference, the genome sequencing and the comparison between the two different subgroups of jejuni found that the genetic changes in this peptidoglycan synthesis genes, PGP1 and PGP2, lead to this shape changes from helical cells to the rod-shaped cells. Well, this figure shows you some main syntheses in peptidoglycan growth. Besides the two proteins that we mentioned in last slides, you can see that there's much more proteins involved in this process. Moreover, remember that bacterial cell growth include both the cell size enlargement and also the cell number increasing with like cell division. So figure C and figure 4 shows you exactly the two processes of cell growth. So you can already notice that there's a lot of different proteins between the two figures. So what I would like to show you here is that the purple structures and in figure C and the green structures in the figure D. So the question is, what are they? Let's again recall some knowledge from one of your previous courses. So as you may still remember that in eukaryote cells, there's also cytoskeletons such as tubulin and actin. Well, bacteria also have this homologs to the structures in these eukaryotes. So as you can see in these slides, the previous proteins that we mentioned, the green color is Fc polymers that which is a homolog to the tubulin in the eukaryote cells, and also the MREB polymers, which consist of MREB proteins. It is a homolog of actin. And for most for the Vibrio and Spirillum, which has more complicated cell structures, they have also another protein that helps them to form these specific shapes. Well, need to mention that these cytoskeleton proteins organize into filamentous structures and play their roles as a protein polymer. So it's not one protein, but a protein polymer. This is an important message that you need to remember. Well, these cytoskeleton proteins actually control the synthesis of bacterial cell wall. As you can see here, the new cell wall are synthesized in multiple sites, as shown in this right circular place. So it happens in multiple sites, not from only one end or from the center. So when the cell grows larger enough, they will start to divide it into two daughter cells, termed cell division. So cell division, it is important to discuss, also because it sometimes can be a drug-targeted fundamental process. So as you can see here, let's see if it works. Okay, so continue. Yes. So most bacterial cells divided by binary fission. So binary fission is splitting of bacterial cells into two cells that are approximately equal size. So cell division starts from as you can see here, start from this cell elongation, and when the cell size start to increasing, then the septum is formed in the middle of the large cells to separate it into the two equal-sized cells. Well, the cytoskeleton controls cell wall synthesis both during the cell elongation and the cell division process. The coiled structure in this photo, the green coiled structure, shows the place of MREB polymers, and you can see that you can see that this coil structure from this fluorescence labeled cells, the green color here is basically all around these long cell shapes. And the phase Z proteins form a ring in the center of the cells. This is a labeled phase Z ring. You can see that the light ring structure formed exactly in the center of the cells. So if everything is working normally, then you can see a range of cell size from small rod of recent developed divided cells to a longer pre-division divisional cells, as you can see from this photo. Here is just after division, and this is just before the division. But what's happened if MREB is blocked? Then you can see that the cells cannot make cell wall during the elongation, and cells become progressively shorter and fatter before they eventually die. And if there's a deletion of phase Z, then the cell, not a deletion, but a block kind of knocked down. Cells can no longer divide it, but they continue to grow longer and forming very long filaments that eventually become unstable, and the cells again lyses and dies. So phase Z is important also because it's involved in several steps in the cell cycle. So the cell cycle normally starts from the replication of a nucleotide, followed by a segregation of the nucleotide. So phase Z plays a role to make sure that the two copies of nucleotide or chromosome locate at the two ends of the cells, not in the same site. So when they forms in the middle of the cells, then they will form in the middle of the cells, as you can see here, and become a ring shape and start to the size could become smaller, and then they just separate the long cells into two daughter cells. So here are some photos showing you the cytoskeleton ring. That's the phase ring that's formed in exactly the center of the cells. This is very start of. This is labeled by some golden particles, and then you can see it. Yeah. Yeah. And phase Z labeled with the fluorescence. So here I would like to quickly sum up the knowledge of this small section. So we talked about the intercellular structures. We know that the cell shape depends on depends on the work of two parts. So the first part is peptidoglycan, and also depends on the cytoskeletons. So the cytoskeletons, so we introduce a little bit on the two important proteins, MREB and phase Z. At last, we talk about the cell divisions and the cell cycles in bacteria.